Well, we're very happy to have Dr. Ard Louis with us today. Art was many years here in Cambridge, and then we finally, I'm afraid to say, lost him to Oxford a couple of years ago, was it? And he's now there, the university lecturer in theoretical physics. And actually, Art is born in the Netherlands, but has had a very international life. You can read his biography in your handout, but he got his PhD in theoretical physics from Cornell University in the United States. And Art has been very much involved in the topic he's going to be talking to us about this morning. In fact, there is a Cambridge Templeton consortium for the emergence of biological complexity, which is basically a grants program funded by the Templeton Foundation. And both our previous speaker, Simon Comey-Morris, and Art Louis, as well as some others, have been very involved in giving out money to others from Cambridge. And that program is still in progress to a number of research groups around the world. And Art actually was responsible for the biochemistry and fine-tuning program in that. So we're still continuing on the same theme then this morning of fine-tuning, this old phrase again. I'm sure we'll keep discussing the meaning of that phrase, but now it's fine-tuning and biological complexity. So thank you, Art.
Thank you very much, Dennis, for inviting me. It's a pleasure to be here back again. So I was also speak about fine-tuning and biological complexity, and I put the fine-tuning in quotes, and I think hopefully you'll see why that is at the end. I'll tell you a bit about myself. So I was here for 8 years in the chemistry department. I just moved back to physics in Oxford, but I'm mainly interested in biological things. The kinds of things I'm interested in with my group is something like this little movie from Keiichi Namba's group in Osaka. It's an animation of the self-assembly of a bacterial flagellum, a flagellar motor, which is a little motor that bacteria use to drive these flagella that they propel themselves with. It's an absolutely amazing piece of engineering. It can spin at a very, very rapid rate, a few hundred hertz or up to 1,000 hertz. And what's most amazing about this to me is really the following, that this thing makes itself. Okay? So, in other words, if you compare this to anything that you'll see in like a car, you go to a factory, it's made an assembly line, or something's made by external things, this thing makes itself, it self-assembles. And I think I first saw this movie and I thought, that is really amazing. I wonder, how does that work? And I think it's very hard not to look at something like this and to ask yourself, how does that work? In fact, the amazing thing, you may not realize this, but you have 10 times as many bacteria in your body as you have cells in your body. It's because they're smaller than your cell. In fact, they do lots of good things in your gut, and you need those bacteria. So as we're speaking, this is happening millions and millions of times inside your body, this very process. In fact, this is a kind of a very beautiful example of it, but it's happening again and again. And the question is, can we understand what the principles that biology uses to self-assemble? How does that work? We'd like to understand that because we'd like to emulate it. We'd like to make some kind of nanotechnology. Anything which is going to be really small will have to self-assemble. We'd like to understand the principles that nature uses to make these very complicated objects, that make these flagella with which the bacteria swim. They twist in a kind of, in this particular case, in a corkscrew type swimming fashion. And of course, one of the problems with this is that this is an incredibly complicated object. In fact, there's a lot that we don't understand about it. And so I actually got interested in this and thought, well, I'll look at something simpler. So one of the things I'm interested in at the moment are viruses, which are quite a bit simpler. So viruses are little caps, basically little things that hold DNA or RNA inside of them. And that's all that they really are. They're little shells with some stuff inside. And they stick to your cells, they inject their RNA or their DNA into you, and then they use the cellular machinery to replicate themselves. And one of the amazing things that was realized back in the 1950s and 60s is you can take some viruses, particularly some DNA viruses, take the DNA out, put them in vitro in a test tube. You can publish in a test tube. You change the salt concentration, you change the pH, something, and they'll fall apart into little bits. In this case, a bunch of identical proteins. You change it back, and they'll reform into exactly the same shape again. And that's, I think, a really amazing feat. Simpler than the one I showed you before, but even that's quite amazing. You take these little balls, you dissolve them, and then just by moving on randomly, if you change the properties back, they'll change right back into the same shape. And so I got really interested in this, and I thought, well, how does this work? What are the principles behind it? And so with my students, postgraduate students, realize the way science works is that our PhD students and postdocs do all the work, and people like myself travel around talking about it. So this is work from one of my students, Ian Johnson, where we tried to model this on a computer. So we said, let's make a very simple model of viruses, in this case little pentagons, with some interactions. And can we make interactions, can we design interactions such that we put these particles on a computer, we let them move around, and it will spontaneously form little capsids like this. And this is one of the first ever simulations on a computer, so they're computer viruses. Really, he's making computer viruses, designing them such that they will design themselves, and they'll make themselves. And sometimes I think this is a little bit like playing with Legos. Okay? And so the great thing about Legos, you can make all kinds of things for them, trains, houses. But I also thought that I could make Legos, which I put them in a bag, I shake it, and out comes a fully formed train. Now that would be a really spectacular toy. I'm sure if I could make a great spin-off company and make lots of money, I can figure out how that works. But, okay, I think that might be a few steps away. But clearly nature uses those kinds of principles, something along that nature, along that style, to make things. And it's really interesting to think about how that works. And so what in my group we're interested in is how does this work? How do these things evolve? What are the principles, design principles, that nature uses to make these kinds of things? And in fact, this principle of self-assembly is it's been around for quite a long time, and it's been studied in quite depth in proteins. So you know, proteins are the kind of molecular workhorses of the cell. They do all the hard work, and so you can think of them as a chain of molecules, polymer. But the amazing thing about them, so they'll float around in space like little balls of string, but then they fold, which means they go into some three-dimensional structure, which would be something like this, particular what's called a native structure. And exact details of how they fold fixes their biological activity. In other words, they have to fold in a particular shape, and then they'll be biologically active. And what's amazing about proteins is you can take the same protein and fold it and refold it, and it'll always find exactly the same final state. And this is something that proteins, a feat that proteins accomplish regularly. And the question a lot of people have is, how does that work? This is partly brought into sharp relief by Cyrus Leventhal in the 1960s, who made the following argument. Let's say I have 150 amino acids, that's 150 different units in my gene. And I then say, okay, each of those could have 10 different conformation angles between each one. Then to first order, I have 10 to the 150 different ways I could fold up that protein. If you take any kind of reasonable time scale for that to move, it would take longer than the age of the universe for that protein to search through all possible spaces. And yet it repeatedly, in a very, very short amount of time, finds this final state. So what's the guiding hand? What's guiding it down? What's making it do this? And there's been a lot of work on this through biologists around, and I have here it's a nice paper by Chris Dobson here in Cambridge. It's a nice review of the idea is that in fact this thing doesn't randomly search through space. That encoded into its sequence is not only the final state that it reaches, but also the pathway to get to that state. Okay, and so that's how it repeatedly finds the same state each time. And in the kind of things that I showed you with these viruses, the reason why we were able to get it to work was because we were not only encoding a final state, which is not so hard, we were also encoding pathways that were easy to reach all this kind of state. And what's interesting about this problem is I think that if you think about it naively, if you didn't actually see viruses support, if you didn't actually see proteins fold, you only found fully folded proteins in nature, and you never observed them fold, nobody would believe that that could happen, that they would somehow from a random beginning configuration always end up in the same configuration. You can make very powerful combinatorial arguments like Cyrus Leventhal's to show that this protein could never possibly find its final state. And yet we see it happen all the time, we see experimentally. So I think that's the thing about biological assembly, it's amazing, but if we didn't observe it, nobody would believe that it was possible. Okay? And we actually know very, very little about how it works, and I would say we're very, very far from any kind of predictive biology where you can even take components and say this is what they're going to make. But we have some ideas about how that works. And so I wanted to show you this partially because I wanted to introduce you in a very hand-waving way into the exciting work that I think I'm doing. I'm amazed that people pay me to play these kinds of games, but that's great. This is one of the luxuries of modern life. You know, a long time ago, not too long ago, to do science like this, you had to be independently wealthy in one way or the other. We have a modern class of people, like myself, who get paid to do these kinds of games, and it's fantastic. But the other lesson I think we get from this is that science surprises us in lots of ways. And I think if this self-assembly, if we didn't observe it, nobody would believe that it was possible, because we have no analogs in our day-to-day life of anything remotely like it. The other thing is that actually we understand so very, very little about how it works. We have some basic ideas, but they may be completely wrong. Right? The idea I showed you with the virus self-assembly may have nothing to do with viruses. We don't even know for sure. But it's an interesting kind of thing. What this also does for a person like myself who's a Christian, and so I'm going to do some of this from a Christian perspective. I realize there are people from other religious backgrounds in the audience, but I think a lot of the ideas I say will carry over without too much difficulty. I think what this does is it gives you a sense of worship as well. I have here a quote from Francis Collins, who's the director of the Human Genome Institute. It's a very nice quote. He says, "The work of a scientist in this project, particularly a scientist who has the joy of also being a Christian, is a work of discovery, which can also be a form of worship. As a scientist, one of the most exhilarating experiences is to learn something that no human has understood before. To have a chance to see the glory of creation, the intricacy of it, the beauty of it, is really an experience not to be matched. Scientists who do not have a personal faith in God also undoubtedly experience the exhilaration of discovery. But to have that joy of discovery mixed together with the joy of worship is truly a powerful moment to a Christian who is a scientist." So now if we start thinking to ourselves, how does religion or faith, Christian faith or whatever faith we have reflect on these things? Well, then what science does in some sense is it expands our ability to see, our telescopes and our microscopes, the kind of beauty of creation. This is a very old idea, and this links to this question of how does God reveal himself through nature? How can we find God by looking at nature? And if you look in the Christian perspective, you can see verses like this very famous passage from Romans, which talks about people being without excuse because somehow aspects of God, his eternal power and divine nature, have been clearly seen being understood from what has been made. This suggests, in some sense, some kind of, you can see something about divine nature by looking at the world around us, is what it suggests. That's one aspect. I think probably the most common way that nature is described in the Bible is as something that brings us to worship. A very well-known passage from the Psalms, the famous poems. It says that the heavens declare the glory of God, the skies proclaim the work of his hand. Day by day they pour forth speech, night after night they display knowledge. Something about the glorious nature of God. Here's another one which is related. When I consider your heavens, the work of your fingers, the moon and the stars, which you have set into place. What is man that you are mindful of him? The Son of Man, that you care for him. This is again suggesting something about our own smallness in the massive world that we're in. If you think about that, astronomers often use that example to say, there are hundreds of millions of billions of stars, enormous numbers, and that gives us a sense of our finiteness. But that's something that people back in ancient times also could have, simply by looking at the stars in the sense of their own smallness compared to the grandeur of nature. And so if I like skiing, and so if I'm in the Alps here in Austria, then somehow there's something about the mountains and their beauty which evokes a kind of reaction, often a religious reaction. It's very interesting to read the book of Francis Collins on the language of God, where he talks about his own coming to faith. And he says that one day he was walking in the Virginia mountains, and he comes around the corner and he sees a frozen waterfall. And he said he says it was a beautiful afternoon, and suddenly the remarkable beauty of creation around me was so overwhelming that I felt I cannot resist this another moment. And he became a Christian. Now the interesting aspect of these mountains and of the frozen waterfall is that we do understand how mountains are formed, right? Mountains are formed by this relentless pressure from underneath, the continuous erosion breaking down from above, and that creates something very beautiful. I don't think Francis Collins thought that the ice of the frozen waterfall was a miracle, a divine intervention of power by God. Nevertheless, there's something about that which evokes our sense of the divine. I think that's an important thing to remember in the kind of things that I'll be saying. So, if we think Christianly about biological complexity, which is probably perhaps a slightly better title for my talk, then I would say the first thing that we get is a kind of sense of wonder and worship, that we're fearfully and wonderfully made. And that's probably not a very controversial thing to take away. I think by and large that's one of the main things we find from the Bible is that those are the kinds of reactions we're expected to get from nature. Other conclusions are more controversial, particularly I think there are many cultural barriers to constructive Christian or religious engagement with science. And I'll talk a few of those cultural barriers if I have time. And particularly the question of origins, which is one of the sub-themes of this talk, is difficult. And I think that's because underlying it is this feeling that where we come from determines who we are and determines how we should then live. I think that's where a lot of the ferocity comes from in these kinds of pictures. And the question is, this is really a theological question, and is that really true? And how is that true? I think also these things have been hijacked by all kinds of ideologies. That's made it a very complex and almost kind of a minefield. So I think one really important thing I want to stress when we look at these kinds of questions in science and religion is to remember that a lot of the problems we have are a little bit like cultural problems. So I was born in Holland, I grew up in Africa, then went to Holland, then went to the US. And when I first came to Britain, about 9 years ago, I had learned my English in the US at the time. I learned there were many differences between the Brits and the Americans. So for example, if you think about culture, people sometimes say culture is like an iceberg. There are aspects that are different, things at the top, the way rich people speak. But there are lots of things under the surface that you don't see, which are much more subtle. And usually when there's a collision, it's these things that are under the surface. So for example, when I first came there, for those of you that are Americans, there are obvious differences. One of them is that these things in the England are called trousers. And the things I'm wearing underneath them are called pants. Do you remember I was just here with a British friend of mine. I thought, I'm in Cambridge, I should buy a nice suit. So I went and found a suit jacket. And the lady, oh, I like that jacket very much. Do you have any pants to go with it? Her face turned bright red. My friend was so offended. He means trousers, he means trousers. Similarly, luckily I was protected from this, but at the time I thought it would be fashionable to wear these things you hold your trousers up with, which are called braces in America, in England, sorry. But I didn't realize that the Americans, they call these things suspenders. That's what a woman uses to hold up her stockings. Right? So I was protected from that. Those are the kind of superficial differences. But I realized when I came here, there are many, many more differences under the surface with my Dutch or my African or my American culture. It's taken me a long time to pick up. And a lot of the kind of conflicts that I sometimes had, I've realized were related to those kinds of under the surface issues. And so I'm going to use this kind of cultural paradigm and come back to it a few times in my talk. So when I was also talking about fine-tuning, I thought maybe I'll take a step back and look at fine-tuning from the culture of physics. This is arguments from physics, and just basically two very popular kinds of arguments towards transcendence, perhaps, that have been used from physics. One is the unreasonable effectiveness of mathematics, and the other one is this question of called fine-tuning in cosmology. And they've been referred to in a number of talks up to now. So let me give you one on the unreasonable effectiveness of mathematics, because this had a big impact on me when I was an undergraduate. And one of the really beautiful stories that John Polkinghorne alluded to yesterday is this question of quantum mechanics and relativity. It happened just here in Cambridge by a very famous physicist, Paul Dirac, at St. John's College. If you go to St. John's, you can still see his room up in one of the turrets. And what Dirac was worried about was the following. You can write down the Schrödinger equation, which had come up in 1926, which is an equation. This is actually, H gives you the energy of a wave function. Don't worry if you don't know what this means, but basically this is the equation that you use to describe small things like atoms. And basically, all of chemistry can be derived from this equation, with a few small exceptions. So, like some heavy element, you need to worry about relativistic correction. But basically, this is what describes all of chemistry. It's a very powerful equation. At the same time, when that was around, people knew about Einstein's special relativity, which is the theory of things that move very quickly. And here you may remember E is mc squared. So this is a slight generalization of E is the square root of mc squared squared. So that just gives you mc squared. And this is the momentum. So if you're moving, this is a generalization of E is mc squared for moving particles. Okay? If you don't moving, your momentum is 0, and then this gives you just E is mc squared. And Dirac was basically saying, well, I would like my Schrödinger equation, the equation that gives me the energy of these small particles, to also work when I make things move very quickly. And it's not too hard to say, okay, you can work out a few small transformations. You can work out that your energy has to follow this formula, and to say, okay, I'm going to make my wave equation, generalize it a little bit, so that it follows it follows this equation. And he did this for electrons. Electrons have a spin up and spin down, so you treat them as two components. But the amazing thing to do is if you write that down, it's not very hard to show that the only equation you can come up with that satisfies this property is an equation with 4 components. And I remember as an undergraduate, I took an advanced quantum mechanics course, and I learned this, and I remember thinking that cannot be true. So I went home and I spent an evening trying to derive an equation without the extra components that would satisfy relativity, but you can't. It's not so hard to do. It's not so hard to show that. That's the only way you can show this. And what's really amazing about this is that the extra components were positrons, or antimatter. They're exactly the opposites of electrons. And although Dirac he said he never had the guts, or never had the courage to predict this was going to happen, in 1932, Carl Anderson discovered positrons in cosmic rays, and they both got the Nobel Prize in 1933. But what's I think really amazing about this is what people often refer to these unreasonable effectiveness in mathematics and natural sciences. Very famous paper by the Nobel Prize winner Eugene Wigner. Is a gift we neither understand nor deserve. Is the following: how is it that you take two very different things, special relativity, which is pretty easy to understand, quantum mechanics, which not so easy to understand, but you can kind of derive it, you put them together, and out comes something like antimatter. When I finally convinced myself that this was the only way of doing it, I couldn't sleep all night. I thought that was the most exciting thing I'd ever learned in my entire life. Because how is that possible? Something so counterintuitive, something so bizarre, comes simply from the mathematics. It's mathematics seems to impose it on us in some very amazing way. And what that kind of brings up is the following kinds of things. Why is the world so mathematical? Why does it follow these kind of beautiful mathematical, reasonably simple mathematical laws? And moreover, why can we understand mathematics? Dirac famously said the equations are smarter than we are, right? Why is the word intelligible in this mathematical sense? Those are very interesting questions. They definitely elicit wonder to those of us that have dabbled in this. And it's really hard, I think, not to, you can think about that in a lot more detail. There's a very nice book by Mark Steiner from the Hebrew University, which looked at this in much more detail, the applicability of mathematics to the philosophical question. And John Polkinghorne has written very eloquently on this thing. But that's something that when you're a physicist, you're trained in. This is part of your culture, your cultural heritage. And it's hard not to be very deeply impressed by the power of these kinds of arguments. Another kind of argument that's used a lot in the science and theology literature is this question of fine-tuning and the anthropic principle in physics. And I have a few quotes which is basically says the universe is the way it is because we are here. It's kind of the universe is fine-tuned for life. It's got particular properties that allow us to be here. One is from Max Tegmark. So there's many, many different ones. If I find structure constant, which change by 1%, the sun would immediately explode. You can come with lots of these kinds of arguments. A nice book on this topic is Just 6 Numbers by Sir Martin Rees, or actually now Lord Rees, who's Master at Trinity College and President of the Royal Society. I'll give you one example, for those of you that are not familiar with these arguments. This is perhaps one of the cuter examples, to give you a feeling for the flavor that these arguments have. And that is a really kind of nice story by another Cambridge man, actually, Sir Fred Hoyle. Fred Hoyle was a very kind of interesting thinker. And one of the things you may not realize is when the Big Bang happened at the beginning, you can get hydrogen, you get helium out of that, but that's about it. You don't really get more heavy chemical elements. And the question people were asking themselves was, where do these heavy elements come from? And the idea was, well, they come from stars. So the sun, for example, is a giant nuclear reactor. Inside the sun are hydrogen atoms. They fuse together to form helium atoms, and that releases energy. Eventually, the hydrogen will be burned up, and you'll have just helium left. Those heliums will fuse together, and there's a whole sequence of these. At some point, this star will explode, and a supernova, it'll spew its dust up through space, and that dust will accumulate into planets, and eventually into you and I. So we're all made, in fact, of stardust, dust of stars. It's kind of a romantic notion. It's a nice, cute idea. And the thing that Hoyle was worried about, Hoyle and Salpeter and a number of other people, was, and Hans Bethe, how does this happen? How does this stellar nucleosynthesis, synthesis of nuclei in the stars happen? And he was particularly interested in how you make carbon. And I'll simplify the story roughly like this. So heliums weigh 4, you can get carbon 12, so you need 3 heliums to make a carbon. That's a fairly rare collision because you need some kind of 3-body collision. And you need the right amount of carbon and not too much oxygen to appear. And people couldn't really figure out how this worked. And Hoyle had a stroke of genius where he said, look, I know how much carbon and how much oxygen I need for life. So I'm going to invert this problem. I'm going to say, I know how much I need for life. I'm going to try to predict something about these collisions. And he worked out there's going to be a resonance, which is a fancy word for an unstable energy level, in the nucleus of carbon. It's an energy level which is not normally used or populated, but in a collision it is populated. And it greatly enhances the probability of these nuclei fusing together. And people didn't really believe it, but Willy Fowler at Caltech eventually had a look, and lo and behold, there was this resonance. Exactly the right energy to allow this stellar nucleosynthesis to happen. And so Hoyle said, would you not say to yourself, some supercalculating intellect must have designed the properties of the carbon atom, otherwise the chance of finding such an atom through the blind forces of nature would be utterly minuscule. Of course you would. A common sense interpretation of the facts suggests that a superintellect is monkey with physics and biology. And he said that his atheism was deeply shaken. So this is the kind of fine-tuning arguments people will make. They'll say, look, there's something really striking about the way the constants of nature are. The nuclear constants are fine-tuned just right so that the nucleus of carbon has an energy level that allows the stellar nucleosynthesis to happen, which is what allows the chemical richness that we see in the world. And that is a fabric upon which to build all of the beautiful complexity of biology. And isn't that amazing? And I think it is quite amazing. And so we can go into a lot of detail about how important these arguments are and how much weight we can put on them. And that's something perhaps more for philosophers to worry about. But I think one important issue is that fine-tuning is not a proof for God in any kind of sense. It does seem more consistent with theism than with atheism. But you could also make the counter, and you can notice the difference with the kind of God of the gaps argument, which is the idea that here you're not kind of saying there was some kind of miracle that happened that we can't explain. We can't explain what happens, at least in the carbon resonance. What's just amazing is that it does happen. And so there's a difference there, I think, with God of the gaps. Although, and here's a nice thing by Martin Rees, he says the following: we seem to have 3 choices. We can dismiss it as happenstance, which means it happens, there we are, nothing we can do about it. We can acclaim it as the workings of providence, which suggests some kind of divine backing behind it, or my preference, we can conjecture that our universe is a specially favored domain in the Sylvester multiverse. This multiverse contains every possible set of laws and conditions, and the existence of our own world and its particular characteristics would be inevitable. And this is a very problematic but interesting idea. Who knows? We don't really know what is going on. I think people have looked at this in much more sophisticated than I do. The question is it providence? Or you could say, well, this is a common thing, objection I get. Well, if it hadn't happened, we wouldn't be around. Okay? And so therefore, we wouldn't be around to worry about it. And so it's a little bit like if somebody throws you a particular deck of cards, the chance of that particular hand coming is very, very small. But that's just the way it is. But it's a bit. I think this carbon resonance and there's many of these other kind of what seem to be coincidences, all very, very surprising. And even if we find a grand unified theory which fixes all the constants, we still have to ask ourselves the question: why are the constants such that a fruitful life could emerge? And it's a very, very interesting question. There's a famous analogy by John Leslie, a Canadian philosopher, the firing squad, who says something that assume that you're up to be executed and 10 or 50 marksmen all point their guns at you, and they say ready, steady, fire. They all shoot, and everyone misses. Okay? You could walk away and say, okay, look, if they hadn't missed, I wouldn't be around. And so I wouldn't be around to worry about it, so sometimes they miss. And sometimes all 50 of them miss, and they'll be around. But that would be a little unsatisfying. I think probably if this happened to you or I, we'd wonder if there was some kind of put-up job. Something happened some reason behind this. And I think that's probably the best we can do with these fine-tuning arguments. But they're very interesting, and they're very fascinating. They're also very useful as a kind of counterweight to a lot of the populist discussions about science and faith, which suggest that somehow the whole weight of science is against anything which suggests the existence of a creator. Now, physics is easy in many ways. It's you can do these kinds of arguments. You can say, well, you can work out what happens if you change the constants. You can do the counterfactuals. Okay. The question is, can you do something like this in biology? So I'm going to give you a quick overview of some kinds of things that people have looked at in biology and try to extract purpose of some kind. And we'll see about how successful that really is. So one of them is look at the history of life on Earth. One of the really interesting things about the history of life on Earth is that it's been so long. Okay, this is 4.5 billion years when the Earth first formed from an accretion disk. We have this late heavy bombardment when the Earth was bombarded by planets, by asteroids again and again that basically vaporized everything, wiped everything out. Not too long afterwards, it seems like we can find the first suggestions of early life. We find some rich carbon-12. The world was dominated by microbes until about 1.5 billion years when the first unicells and multicells came, and then man at the very end. And what you may not realize is if you think about the time, put this in perspective, let's say I make this history of the Earth a 24-hour day. Okay. Then, basically animals, which is around here, the Cambrian explosion, didn't appear until the last 2 hours of the day, so at 10 o'clock at night. Dinosaurs went extinct just 20 minutes ago. Okay. Anatomically modern humans were only appeared in the last 1 or 2 seconds. Recorded history is probably the last 1/10 of a second, and your own life is probably the last milliseconds of the day. And that gives you some perspective on how much time we've actually been around. And you could say, this is very similar to the passages in the Psalms which look at the heavens and say, look at space, how big and glorious it is. Doesn't that point us somewhat towards the greatness of the Creator? You could say, looking at time in the same sense, gives us an impression of grandeur. And that's one way, I think, very straightforward way of looking at things. Of course, there's this thing about the late heavy bombardment, first fossils, why we think we may have found life. Basically, it seems like life emerged quite rapidly after this first heavy bombardment. So, there's perhaps this question I said about the age of the Earth, the age of the universe, giving us some sense of the grandeur of God. Another very popular thing that people have looked at, and which I spoke about last year at the Faraday Institute in much more detail, was the origin of life. How did life originate? Perhaps we can look at that and find some signs of God in one way or the other. A lot of people have wanted to do that. The question is, what happened here? How did life emerge, the very first kind of life? And first you have to ask yourself the question, what is life? And there's a famous American judge called Judge Potter Stewart who was asked in the 60s to distinguish between pornography and art. And he said, well, I can't write down a description, but I know it when I see it. And there's a sense in life sometimes that if you want to say, what is life? There's a little bit of that to it. A very popular definition by Gerald Joyce from the Scripps, this is from the NASA exobiology program, that life is a self-sustaining chemical system capable of undergoing Darwinian evolution. And roughly speaking, life is something which has replication, which can copy versions of itself with some fidelity, and it has some kind of metabolism, which means it can take energy from its surroundings and live. And so we'd say viruses are not really completely alive because they need something else to do this. They need some hosts to do their replication, in particular their metabolism. So one way of thinking about what is life is to think, well, what's the minimal possible life you can have? One option is to try kind of a top-down approach, where you take the very simplest organism, keep knocking out genes. And there's a big program at the moment by Craig Venter and others trying to make synthetic life. Craig Venter is a chap who was competing with the Human Genome Project to sequence the first human genome, a very bright and enterprising character. He's taking Mycoplasma genitalium, one of the simplest organisms, and basically knocking out as many genes as possible to see what the smallest possible life form you could make. And you can get down, it's argued about how many genes, on the order of 300 genes, and then the thing basically peters out. It's still a very complex organism, extremely complex compared to anything kind of chemical. So that's probably not the best way to understand what life is. Perhaps a better way, or maybe it is, perhaps a better way is try to do some kind of bottom-up approach to life. This brings us back to Charles Darwin, who said if, and oh, what a big if, we could conceive in some warm little ponds with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc., that a protein compound is chemically ready to undergo still more complex changes. And so there's been a long history of people thinking about this kind of primordial soup. It's back to the Russian Alexander Oparin and J.V.S. Haldane here in the UK, who came up with this idea that perhaps you could have chemical systems that could duplicate themselves, that could have both some kind of proto-metabolism or some kind of proto-replication. And probably the most iconic example of that was this very famous experiment by Miller and Urey in 1953. Stanley Miller, a young 23-year-old graduate student in the lab of the famous Nobel Prize winner Urey, put some sparks through this chamber, a vapor of water, ammonia, methane, and hydrogen, and he produced amino acids like glycine, and he produced some sugars, nucleic acids, all the basic building blocks of life. That was a very exciting experiment because it seems quite simple. You just put sparks through some simple chemicals and out come these very complicated biopolymers. And this is often taught as this is the way you can understand the origin of life. And definitely very, very striking, there's no question about that. There's problems with that experiment, that particular experiment. One is that the atmosphere that he used is probably not the same as the atmosphere of the early universe. It's also very hard to make particular components, particularly things like the things that you use to make RNA, like ribose, is very, very unstable. And also mainly these experiments make tar, not a very high yield of good things. Nevertheless, there's been a very long history of working at this, and I like this quote by Leslie Orgel, one of the great men of Origin of Life, who was also in Cambridge for quite a while before he went to I think, Scripps. He said, it's hard to believe that the ease with which sugars, amino acids, purines, and pyrimidines are formed under reducing atmospheric conditions is either a coincidence or a false clue planted by a malicious creator. And it's very striking, there's no question about it. There's others, this is Gunther Wachterhauer, who has a kind of a competing theory for the origin of life. And he says, as far as I'm concerned, the soup theory is more a myth than a theory because it doesn't explain anything. So this is an example of the kinds of views you can have in the literature. And the problem really is that you do find it not so hard to make some of these simple chemicals, but to make the real biopolymers like DNA or RNA or protein is much harder. And basically anywhere where you get energy in simple carbon-rich molecules, you find a suite of interesting you get organic molecules. So I said Stanley Miller empire, he died just in May, unfortunately. He had a big empire around him, people trying, at least from his perspective. People also can form molecules under high pressure. A lot of these molecules will form under high pressure conditions. You can make them on hot rocks and on clays. You can find them in giant impacts. You can find them in hot deep sea vents. There are ideas that they come from outer space. So you can look at comets. You can often have certain amino acids and some lipids on them. Glycine has been found in outer space. Spectroscopy methods. So lots of different ways of looking at these things. Miller was quite a character. And he said, you can find lots of nice quotes from him. So he thought the following about organics from outer space. That's garbage. It really is. So there's no shortage of strong opinions. But the difficulty is really making these biopolymers. And Miller said this, we understand pretty well how to make them, but then you have to make first self-replicating polymers. And actually nobody really knows how it's done. So that was a quick whistle-top tour through some of the very early problems of chemical evolution. It seems that life emerged rather quickly after the early heavy bombardments. And the question is, why did it emerge so quickly? How did it emerge so quickly? There's lots of different theories of chemical evolution, which are different ways of making chemicals. There are definitely a lot of surprises, but it's also lots of things which are surprisingly hard to do, particularly making biopolymers. And then from there, you've got to make a lot more steps. You've got to go to some kind of system which has replication and metabolism. There's a theory of an RNA world, a world of RNA molecules, which can be both catalysts and information stores. Still probably very schematic, but there are interesting fossil relics, like the fact that our ribosome, the object that makes proteins, has lots of RNA scaffolding in it, suggests that perhaps it's some remnant of an earlier RNA world. And there are basically two camps. roughly speaking. One is the camp that says life is ubiquitous, so life emerged really rapidly after the early heavy bombardment because life can easily emerge. And a lot of astrobiologists would be in this camp, that you're going to find it on lots of other planets. It's going to happen again and again. You might even find life in other solvents. You might find life in ammonia or who knows what. The other view is that life is a near miracle. It happened through a very special combination of circumstances in the early Earth, and that will very unlikely ever be replicated. And that's just happened. It just happened, and there we are. And in fact, we don't really know. And another nice quote by Orgel: "The problem of the origin of life has much in common with a well-constructed detective story. There is no shortage of clues pointing to the way in which the crime, the contamination of the pristine environment of the early Earth, was committed. On the contrary, there are far too many clues and far too many suspects. It would be hard to find two investigators who agree on even the broad outline of events." So that's a summary from 1998, but it's probably still true. And there are lots of very interesting ideas, lots of surprises that come up. But as far as I can see, there's nothing really compelling that shows that we understand how life originated. And it could be that something comes completely from left field. Now, one very common response to this question, this difficulty of understanding the origin of life, has been one of this type. And I was here not long ago and I heard this guy, Stephen Meyer, who's a fellow of the Discovery Institute, who said the following. He said, well, okay, look, I've studied this origin of life literature, and I asked myself, could it have happened by chance? Which basically means by some fluke, or could it have happened by necessity, which means life is ubiquitous? He says, well, I can see that it couldn't have happened by either of those, and so I'll use inference as the best explanation, which is an idea in the philosophy of science from Gilbert Hartman. It's been popularized a lot by Peter Lipton here in Cambridge. A very nice book on the best way of looking at evidence and finding some inference from it. And he said the inference is almost conclusively that a designer did it. It's done by God in one way or the other. And this is an argument I've had repeated to me many, many times in many forms. And perhaps, it was a miracle by God, some divine intervention, which is the way a Christian would look at it. God came and implanted life on earth. The problem really is, how would we know that that is or isn't the case? And I think the real reaction of a lot of scientists like myself is, what about surprises? Right? And what about the size of chemical space? Here's a nice quote by Chris Dobson saying, how big is chemical space? Well, chemical space, which encompasses all possible small organic molecules, is vast. So vast, in fact, that so far only a tiny fraction of it has been explored. And the difficulty with making these kinds of inferences and saying, well, it could only have happened by divine intervention, something like the origin of life, is that to do that, you have to show that there's no other way for this to happen. You have to do the counterfactual and say this is the only way it could happen was by some fiat miracle. The difficulty is that the counterfactual space, the space of all possibilities, is far, far too large for us to explore. And the possibility of a surprise is always looming around the corner. So probably we'll never know. And it may not be a very fruitful place to look for signs of intervention, simply because the real the best summary of the field is that it's a vast, warm pond of ignorance. That brings me to another point, which is that a lot of the kind of in the popular imagination, and in fact in the populist literature, the idea is that God's action in the world can be described primarily in this way. You've got something complicated, then a miracle occurs, the origin of life, the origin of the flagellar motor, and then we can go on with understanding how science works. I probably really was a bit unfair pulling him in, but there we go. It couldn't have happened by quote-unquote natural means. And Meyer's inference is one of this type of an argument. And the question really is that the right way of looking at it? Is God only present in the interventions? We heard John de Broek yesterday saying that this idea of Deus ex machina, the idea of God as only being present in interventions, is a dangerous theological idea. But I think it's very common in how we look scientifically. Or we're to think about God somehow being present in the whole thing. The Christian idea of God sustaining the world by the word of his power. And these are ideas that go back a long time ago to Augustine and other people saying we're worried a lot about God's special action and his regular action. You can see this throughout the literature. And so I, in a kind of crude way, like to say, well, a lot of the natural laws, the things I find in science, what I'm really studying are the customs of the creature, the ordinary way that the creature sustains the world. And I believe that there could be miracles where God working in some kind of uncustomary way. I'd say the resurrection would be, of Jesus Christ would be a very clear example of that. But whenever they happen, they always happen for some kind of theological purpose. There's a reason for them. They're not magic tricks to somehow surprise us in one way or the other. They're in some kind of redemptive theological purpose. That purpose may be obscure to us, okay, but that's why they happen. I think that's an important thing to keep in your mind when you're looking at nature and trying to extract some kind of teleology from it. In the last 15 minutes of my talk, I want to look back again at this question of language and say, can we look at the origin of biological complexity, and can we look at the way the language is being used? Can we find forms of language that would be less offensive to people around us? And might that help us to look at it in a different kind of a way? This is a very beautiful quote in Charles Darwin's book, The Origin of Species. There is a grandeur in this view of life, with its several powers, having been originally breathed by the Creator into a few forms, or into one. And that, whilst this planet has gone circling on according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been made and are being evolved. So the question is, if we look at evolution particularly, is this something very beautiful? Are we seeing endless forms most beautiful? This is actually in the second edition of his book, came out shortly after the first edition. He added this sentence, or the reference to the Creator. Now I want to make a little aside and define what I mean by evolution, precisely because evolution has become encrusted with all kinds of competing ideologies. I have a little boat encrusted with barnacles, and so it's important when I use that word to say exactly what I mean. And roughly speaking, I have this kind of typology. I can use the word evolution to mean natural history. The idea that the world is old, the idea that complex forms came from simpler forms, simpler forms precede complex forms. I can also look at evolution as a mechanism for the emergence of biological complexity, the idea of mutations and natural selections as a way of generating biological complexity. And that can be another way of looking at evolution. I can use it as a big picture worldview. So the idea that, famous quote by George Gaylord Simpson, man is the result of a purposeless and materialistic process that did not have him in mind. He was not planned. He is a state of matter, a form of life, a sort of animal, and a species of the order primates, akin nearly or remotely to all of life, and indeed to all that is material, which is a deeply theological statement. And I think what happens very often is when people are in the common public are reacting against evolution, they're reacting against these kinds of statements. Often what they do is then try to attack either natural history or the mechanism of evolution. But I think the connection between there and there is not nearly as strong as someone like Gaylord Simpson or Richard Dawkins would have you believe. And so I'm going to look at that. And so I'm going to talk about evolution now. I'm going to be mainly asking myself, how about these mechanisms? Is there something about them which might be interesting or beautiful? So the first language question I'm going to look at is random or stochastic. So we speak about random mutations and natural selection. And you'll see throughout the literature, and in fact we saw this with Charles Hodge in the talk yesterday, who objected to the word random, the idea of things being random. I think a lot of that has to do with our worries. The word random is very slippery, very difficult to say words. But in my fields, I use Monte Carlo simulations. That's the word for simulations that use random numbers to generate new states that we then select on. This is basically how Monte Carlo works. In fact, your stock portfolio or the interest rate in your bank is fixed by the currency markers, which are by and large calculated by Monte Carlo techniques. And these are stochastic optimizers. And the reason for that is that whenever you're looking at a very high dimensional space, a space with many, many components, very often stochastic optimization is by far the most efficient way of optimizing, the most efficient way of finding new states. And so in these simulations here, we're using a stochastic process to make these things come together. And so if I instead of using random mutations, I would say evolution is a stochastic optimizer in some very, very high dimensional space, then perhaps it would be more palatable to a lot of people who worry about the connotations of the word random. But that's really the way the word random is meant scientifically. It's meant in the sense of a stochastic optimizer. We just looked at this question of contingency and deep structures between Simon and Stephen Jay Gould, so I'll skip that. So again, an example of differences, nevertheless, in how two scientists can interpret natural history in particular, with very, very different kinds of teleological conclusions. And again, it's important to realize that there is this flexibility there in how you could interpret this. Another thing about language, which I think is very important, is gene language. So I have here two quotes, one very famous quote, one somewhat less famous, but I think very interesting. The first is by Richard Dawkins from his book, The Selfish Gene. He says, genes swarm in huge colonies, safe inside gigantic lumbering robots, sealed off from the outside world, communicating with it by tortuous indirect routes, manipulating it by remote control. They are in you and me. They created us, body and mind. And their preservation is the ultimate rationale for our existence. Now, I think he meant this as a kind of metaphor, but it's been widely used in a kind of sense of genetic determinism almost, and it's had all kinds of repercussions. And people have resisted this for a number of reasons, because it seems like a kind of statement of materialism. I think Dawkins himself has worried about how this idea, the selfish gene, this metaphor has been used. But metaphors are important because they are how we understand and how we explain. But here's a book from another Oxford colleague of Richard. In fact, Dennis Noble, who's very interested in systems biology, very famous physiologist. It's a very nice little book called The Music of Life, Biology Beyond the Genome. In fact, Dennis was Richard's PhD examiner, which is one of the small world things. And Dennis has, on purpose in his book, he's interested in connections, in systems biology, in the way that genes are connected to each other. And he says, well, let me take that same sentence and turn it around. It's not quite as elegant as Richard Dawkins almost matched his prose, but it's still nice to read. It says, genes are trapped in huge colonies, locked inside highly intelligent beings, molded by the outside world. And you can see this kind of parallelism, communicating with it by complex processes. And it ends by saying, we are the ultimate rationale for their existence. And he asks Richard Dawkins, is there a scientific argument you could make to make a distinction between this way and this way of looking at genes? And there isn't really. There are two different interpretations, philosophical interpretations of the biological, of the way that biological replication basically happens. I think that's a nice example, again, of the way we can look at the language, the metaphors that we use in biology, and how they can be misappropriated or they can be misleading. Another interesting kind of metaphor in a lot of, I think, in the very common in the public minds is what can you do with biology? What directions can you go? There's an idea of is evolution more like Lego blocks, which means you can build things in certain ways, or is it more like plasticine or Play-Doh or clay? You could make it in almost any possible way. It's an analogy. This is an image I got from Mark Kirshner, and he says, well, one of the nice things that you may not realize about biology is that there are in fact all of these beautiful building blocks from the field of evolution and development, or evo-devo. One very popular one is a gene called PAX6, which is involved in making eyes, or it's a switch for making eyes. You can take the PAX6 from a human, you can put it into a fly, at the bit of the fly which will make wings, and the fly will try to make a fly eye on its wing using a human gene. This is an idea of it's a little Lego block, if you wish, that can be used. And there's a whole series of these Lego blocks. They've got funny names like Sonic Hedgehog, Shaven Baby, because the embryo loses some of its outer layers. One called Tin Man, it's related to heart development. This is from The Wizard of Oz. You know, Tin Men who have no heart. It's called Tin Man. It's a very nice book by Sean Carroll called Endless Forms of the Most Beautiful, The New Science of Evo-Devo and the Making of the Animal Kingdom. And one of the ideas that's floating around is that a lot of evolution happens through recombination of these kinds of Lego blocks. That a lot of the power of evolution is not just by making new proteins, but by reusing proteins in different orders. And I think that's a very beautiful picture. It's a much more elegant picture than the kind of way things are sometimes brought across. And it's not something which is very well understood in the general public. One kind of side effect, I think, to be really honest, a lot of this is still early days. We're still trying to understand a lot of it. But the idea that a lot of evolution happens predominantly through rechanging the wiring of these things rather than through point mutations and changing proteins, I think is what seems very likely to be. Another amusing thing about gene language is actually the question, why are there so few genes? So here I've got a number of different organisms. So Mycoplasma genitalium is perhaps the simplest, the smallest genome. E. coli is bacterium that's very used a lot. This is yeast. It's got 5,400. Yeast got 5,800. Drosophilus is a fly, a fruit fly. It's very common, used in genetics. So 13,000 genes. Here are two little worms. One is C. elegans, which has about 950 cells. It's a very important model organism. In fact, Sidney Brenner and Sir John Sulston here at the LMB got the Nobel Prize for this in 2002. And here's a very similar worm called P. pacificus. It's worked by Ralph Sommer in Tübingen, for example. It's very, very similar. These are organisms that have on the order of less than 1,000 cells. You can watch the development very, very carefully. But this one has 29,000 genes, and this one has 19,500 genes. The question is, why is there a big difference in genes between these two different organisms? And here's my niece. And I guess why does she only have on the order of 23,000 genes? When the Human Genome Project first started, people estimated there'd be anywhere from 100 to 160,000 genes. That's roughly the number of proteins that we have in our body. And one of the surprises was how little genes there actually were. And as we keep looking at them, we find that there's less and less and less of these genes. Moreover, these genes, which are the bits of the DNA which encode proteins, we share a lot of them with E. coli, with yeast, with frogs, with chimps. The question is, what makes the difference? Actually, myself and the chimpanzee. When I was a child, I lived in Africa. We had a chimpanzee as a pet. And the question is, what makes us different? Was I even very different? It's not entirely clear. But what makes us different if we share so much of our genes? And the idea is that a lot of the differences between us have to do with the interactions between our genes. This is a picture of a gene network in E. coli. So each of these little dots is a gene, and the line means this gene tells that gene to turn on or to turn off. So it's called a transcription network. So these are like little switches, although they're analog switches that are susceptible to noise. They're very complicated objects. And this is I think it's about 30% of the full transcription network in E. coli. And people are now studying these in great detail, and particularly a lot of physicists, physical scientists, computer scientists are moving into biology, looking at these objects, because they turn out that something like this network is highly structured. In fact, you can do some very simple calculations of what a random network would be with the same number of nodes, about 400 nodes and about 500 links, and you'll find that these nodes are very heavily biased toward particular types of motifs. And in fact, those motifs are highly convergent, so you can find that the same motifs occur again and again and again throughout nature. You can find all kinds of evidence of evolutionary convergence of these motifs. And you can pick them out and try to study them and say what they do, and they have all kinds of interesting switch-like properties. And some of them they protect you against fluctuation, some of them allow things to happen very quickly, some of them slow things down. And we have the feeling that we can start to untangle the way that this is an E. coli, which is a bacterium, way it thinks, that uses this gene network as a way of thinking. It says if there's some stimulus, some sugar that comes in, it'll react through its gene network, and there'll be a response, which is swim or don't swim. And so one of the things that a lot of us are interested in is how these gene networks work, and how do they evolve? And it seems like these genes are using some kind of modular structure, some kind of almost engineering type modules that they can really quickly switch around, and they can adapt very quickly and evolve very quickly. So there's been a lot of talk about this. I think probably it's a bit of hand-waving sometimes. There was a nice series in Nature earlier this year, little one-page essays. And here's one by Nigel Goldenfeld, who's a physicist, and Carl Woese, very famous biologists, famous particularly for discovering that we have 3 different basic forms of life: archaea, bacteria, and eukaryotes. And they said the following: we believe the molecular reductionism that dominated 20th century biology will be superseded by an interdisciplinary approach that embraces collective phenomena. This is a kind of a bold statement, but it's based on the kinds of advances that we're making in systems biology. The idea is that these connections are what explain a lot of the complexity that we see, and that perhaps one day we'll be able to understand some of this. Now, I should say that this is a real clash of cultures. I highly recommend work by Evelyn Fox Keller. She's a very controversial writer sometimes, but she's a very astute commentator, particularly on this clash of cultures. It's a very nice book that she has. It's called, I'm blanking on the name of the title of the book. It's called, it came out in 2002 in HQP. And she says, does biology have laws on its own that are universally applicable? Or are the physical sciences the exclusive domain of those laws? By its very nature, life is both contingent and particular. Each organism the product of eons of tinkering, building on what had been accumulated over the course of a particular evolutionary trajectory. To what extent will physicists' focus on biology demand a shift in epistemological goals, or even the abandonment of the traditional holy grail of universal laws? And one of the really interesting things that people like myself and a very large cohort of physical scientists moving into biology is we're finding a lot of these kind of cultural differences. They can actually lead to very, very heated discussion and disagreements, which are based on different cultures. We're coming from a culture where we believe in the power of mathematics. We believe in that somehow there are, even in all the messiness, some kind of overarching principles that we might be able to extract. And a kind of biology is often trained in this idea of life being contingent, that each particular mechanism is specific and has a particular history to it. That's crudely speaking. That's roughly what we're running into. And these are very nice paper as well in that same series by Uri Alon, who says there seems to be a degree of simplicity in aspects of these networks, which is intriguing given that cells have evolved to survive and not for scientists to understand. And one of the ideas that a lot of us are now toying with is the idea that we can use principles like robustness, the fact that cells have to be robust to external fluctuations, a certain kind of economy, the economies of replicating, there's separations of timescales, and particularly that there's questions about evolution and convergence that will suggest a certain simplicity that we hope to discover in biology. And if we don't have that, then perhaps we'll never be able to understand biology at the level of an entire cell, or at the level of an entire organism. So those are, I think, fair to say, very speculative ideas. If I say I believe these ideas, I realize that I believe them partially because I'm culturally biased by my physical science background, and I may be completely wrong. But it's fun to at least try to go down this direction and see where we go. Perhaps 50 years from now, we'll have a lot better feeling for some of these principles, whether they do or don't have explanatory or even predictive power. And so the last question is, will biology become law-like one day? Will it become more like physics? Who knows? Maybe it will, maybe it will. Maybe we'll have to come up with completely new concepts and ways of thinking about things. Surely, though, I think we will find new emergent principles which will be beautiful and elegant. And I think will make the whole idea of evolution seem much more beautiful and perhaps palatable to the general public. It's easier to popularize. But it's really too early to tell. And I think deriving meaning from these mechanisms, whichever way you want to go, is still too premature. We just don't know enough about how biology works. We don't know about these general laws, if they even exist, to be able to do the kinds of counterfactual arguments you would need to do to make any kinds of arguments similar to the fine-tuning ones. I think it's going to be very hard to make. And on that note, I'll end my talk. Thank you very much.
Thanks very much, Art. Yeah, Ken.
It's not a question. I just wanted to add an enthusiastic underline to your last two comments in the quotes and papers in Nature. I'm sure you know about the ENCODE project, which is a project that has tried to look very intensively at a relatively small area of the human genome to see how much of it is actually transcribed into RNA. Now, we know very little of any region of the genome is actually consists of genes as we traditionally know them. But the ENCODE project simply tried to look at how much of the DNA was transcribed into RNA, even if it was never translated into protein. Now, typically, we would say 1 to 2% of the human genome is transcribed based on the production of proteins. The ENCODE project showed that more than 2/3 of the DNA sequences in the intensively studied area were actually transcribed, which is like, oh my goodness. And it tells us that there is a level of collective interaction, exactly the way that you described here, that is 2 orders of magnitude perhaps greater than anyone in molecular biology had suspected up until this year. So the notion that there's still surprises Absolutely.
That's amazing. I was completely blown away by that work.
Yeah.
It's incredible. So, who knows what it means, but it's a very exciting time to be in this field.
Some of that, I suppose, can be accidental run-on transcription. So, that it may just be kind of low-level noise that actually doesn't mean anything at all. Just to, put that in context.
That's a possibility, and one can look into the techniques that were used in the ENCODE project as hypersensitive to try to pick up those things, but the details are still to be sorted out.
Good. Yeah, other questions? Yes, fair enough.
Why did you use the term fine-tuning in your title?
Because I was asked to use that term. that was what I was asked to speak about. Simon and I both, that's why we have to.
I mean, this is precisely the opposite of fine-tuning, the story you told.
It is. So fine-tuning is a very complicated word.
Well, not that. It's not so complicated that you can work it into that story.
Into this.
The story you told, which is a wonderful story. I mean, I liked it. It's just that I think that to suggest the metaphor of fine-tuning in the context of biology, I think is basically misleading, actually, because in point of fact, it's the difference between that and fine-tuning. See, in the case of fine-tuning in physics, what's surprising is that you need a very precise starting point to get to where we are. What's surprising, particularly if you take Simon's view of convergence, what's striking in biology is that you don't need a very precise starting point to get to where we are. It seems as though, as you pointed out, that you really don't need to specify the initial conditions in the sense in which only a very precise set of the initial conditions would work. Now, you might want to argue that you have to have certain kind of conditions on Earth in order, etc., but that goes back to fine-tuning in physics.
So I just used the term in quotes for that reason. I think fine-tuning often, when it's used loosely, people are really saying anthropic principles. So they're saying there's some kind of anthropic principle in biology. And what I'm arguing is that the kinds of thinking, so I agree with you completely. I'm saying the kinds of arguments that one makes in physics don't translate into biology. I want to give you one example, just to reinforce your point, perhaps, which is the following. People wonder sometimes whether water is special for life, that you have to have water for life. Could you have life in ammonia? Could you have life in fact, organic chemists do most of the experiments in other solvents, so perhaps we don't really know. Life, obviously, but you can find that, biology is very finely tuned for life. It's very heavily adapted. Biology is very finely tuned to water, very heavily adapted to water. That may just be because it's adapted to water, or is it because water is rich enough? And it's a very interesting question, because if water is, you have to follow the water, which is the NASA idea to find life, then that suggests it's the only possible solvent. And so one thing that people said is, well, look, you can take H2O and replace it by D2O, where you replace the hydrogen by deuterium. So chemically, it's the same object. But for like a lot of bacteria, it's poisonous. They die. Okay, so that's just, life is very fine-tuned to water. This is the someone said to me. Because if you change it just slightly, the bacteria die. However, you can take the bacteria and wean them on increasing amounts of D2O. So eventually, they're very happy in D2O. They evolve to live in D2O. And therefore, H2O is poisonous. So then if you start asking yourself, is there something special about water, you've got to broaden your thinking and say, what is the robustness of evolutionary mechanisms? What is the robustness of the chemical things that are in life? What is the extent to which you could stretch that and still have something as rich as life? And that's a very difficult question to answer. But if you could, then perhaps you could go back and make some kind of anthropic arguments, but it'd be very difficult, right? But there is some analogy between the fine-tuning in physics and the anthropic principle in biology, if you could solve that problem, in the sense that they're both suggesting that there's something very special about water.
The initial conditions?
About the initial conditions, yeah, well, and about the...
But of course that would come back to how we have water in the first place.
Yeah.
So we get back to fine-tuning in physics then.
That's right. And it always falls back to the initial fine-tuning in physics, but it would extend those arguments, if you wish, to say that not only does the fine structure constant have to be right to allow the sun to exist, but also something about the quantum mechanics has to be right to allow water to have the right bond angle, etc., etc., etc. Those are the kinds of, that's the kind of thing you might be able to answer. I just think those questions are extremely difficult to answer without looking at these bigger questions of robustness.
Okay, yes.
Yeah, from Simon's talk and your talk, may I suggest that the fine-tuning is, in a sense, there's a deeper level of some kind of biological natural law, so that no matter what kind of evolution process you take, you will still get that kind of result. So that level of that kind of a natural law, biological natural law, is so fine-tuned so that it's environmentally resistant. You will still get the same answer.
Yeah. We're saying something I think slightly different. Perhaps I think I would agree with Erna that the word fine-tuning perhaps is not the right word to use in this context, because it does suggest a radio which is being very finely tuned, and I think in physics it's slightly different. What I think you're saying, and what Simon speaks about the idea of deep structures, and saying it's somehow there's some kind of biological hyperspace that channels things along certain directions, not along others.
How about say exquisitely designed?
Yeah.
I know people are afraid of saying that kind of word.
Well, the word design is that I think I agree with Simon, which is that the word design has lots of barnacles encrusted on it, and it's not clear. I mean, perhaps most of the theologians would be better at getting a cleaner word.
Okay, other questions?
I'm not trying to be sort of obtuse, but I'm not clear about what the biological fine-tuning means in the manner people are talking about it. Did Simon indicate that from a wide variety of evolutionary possibilities, there's a kind of a narrowing, a fine-tuning in the sense of a narrowing down to a limited number of evolutionary outcomes which have occurred in the real world, or am I off the track with this? Could someone possibly enlighten me, or maybe not?
Yeah, just find something. So, I don't want to speak too much detail for Simon, but I think what he was saying was...
It seems to be looking at it from the other direction somehow. Physics sort of opening out from a small number of initial kind of constants. And I could be mistaken. My impression with biology was possibly it was going down from a wide number of evolutionary possibilities to a narrow number of actual real outcomes. So I'm slightly lost between the two concepts, given I've got a reasonable understanding of what people are talking about.
Sure, yeah. So I think don't want to speak too much for Simon.
Yeah, I understand that.
Himself,
Sure, yeah.
I think this is a quote from the end of this book. So when you examine the tapestry of evolution, you see the same patterns emerging over and over again. So Gould's idea of relative of life is not hypothetical; it's happening all around us. It's evolutionary convergence. Okay. When convergence is the rule, you can rerun the table of life as often as you like, and the outcome will be much the same. Convergence means that life is not only predictable at a basic level; it also has a direction. It's quite a controversial thing to say. I think what Simon is trying to argue is that although we don't necessarily understand why these things happen, in some cases we can. If you look at fish and sharks and things have converged in very similar shapes, you can do the hydrodynamic calculation, explains to you why they have the shape that they do. In other cases, like the camera eye or the canine teeth, it's harder to know why those things evolve in the same direction. I think he's basically making the observational point that we do see things evolving again and again. That's suggestive of somehow evolution ending up in the same direction. If you were to rerun it again, perhaps it would take an extra billion years, or who knows what, but eventually something which is sentient, like ourselves, and self-conscious, and able to interact with God, would emerge. And what he's arguing is that he thinks if he looks at biology, he can see that. And that's a very interesting idea. I mean, a theologian could also say,
could follow Gould and say, if something really special hadn't happened, then we wouldn't be around. So that shows us the providence of God who guided it at just the right time. Right? And you could probably make an argument either way. So I want to take it a little bit lightly and say that it's one or the other. But I think it's very interesting, Simon's argument, certainly. And certainly he would say they do seem to show some signs of transcendence. What is interesting, I think, is simply that people can look at the data and come up with very different conclusions, which I think is an interesting point as well.
Another way of looking at it is just think of all the ecological niches in the world, every little ecological niche that you have. And basically they fill up with stuff that's got to look fairly similar or be fairly similar, because they have to survive and reproduce successfully in that particular ecological niche. So in a way, the planet itself is channeling, if you imagine it as a great matrix of lots of little bits of design space, then everything has to sort of follow down these little bits of design space, because that's the only way you can function successfully in that particular ecological niche. So that's it's saying the same thing but with slightly different words, which is why you get the marsupials and looking very similar to the similar animals in Africa and so forth that Simon was illustrating, because they have the same ecological niches in different continents. So they've
It was the use of the term fine-tuning being superimposed on what appeared to me to be two rather different examples, which I was finding confusing.
And now I'm getting more and more sorry that I even used the word at the beginning, even in the quote. I should have just crossed it out.
Maybe the titles of the talks were made up by cosmologists. Could it be?
I do hope people felt they were able to change the titles rather than stick to the script.
That's fine. You just put a question mark at the end.
Right.
So, okay, now goes yes.
Kind of more struck between the kind of the juxtaposition of this robustness, which is that. Do you interpret that to mean there's a variety of possibilities here, versus this convergence idea of directionality, and to whatever degree there might be this arc, has there been any speculation within evolution as to, I guess I'm back to the factors that cause us? Are they more inherent to the organism themselves, or do they more spring forth from the environment? It's a combination of the two that maybe.
Well, this is a big debate among people. So robustness has a number of different meanings. One meaning can be a really simple one, which is that an organism has a lot of processes in biology have to be robust to fluctuations in temperature, in salt concentration. And so like those networks, we think we can understand sometimes why they converge on certain things and not on others by showing that these are much more robust to fluctuations. And that can explain why they're chosen over others. So that's a principle which we think is more generally useful. The robustness in evolution is often used in the sense of saying something is robust to genetic variation. So you can have mutations in an organism that don't actually change its phenotype in any kind of bad way. Sometimes there's an idea of even evolutionary capacitance, which is the idea that you can build up mutations that are kind of latent, and then in certain conditions they can be released. So a very famous example of that is Susan Lindquist's work on HSP90, which is a chaperone, something that it's a molecule that basically prevents these mutations from expressing themselves. But when you come under stress, under heat shock, then they do allow a lot of mutations to come out. And one of the ideas would be that you've got a robustness to genetic variation inside of yourself, which builds up a capacitance, where if you go under stress, suddenly then you can really quickly modify yourself, even though normally it's bad for you. But if suddenly one of your modifications allows you to adapt to that stress, then that'll take you in a new direction. That's a different kind of robustness argument. That makes sense. And the idea is that you could also look at this and say, well, these are collective effects in evolution in some vague way, some analogical way. And probably those kinds of collective effects, mechanisms are, well, they might be present, they might give it a lot more power. But these are, I should say, these are controversial issues about which people argue a lot.
Okay, any other questions or maybe, yeah?
So the only way I think of doing this is actually a quick diagram, if you want to look at it. But basically looking at this idea we've got of there being these two like conflicting ideas of either fine-tuning giving rise to biodiversity or biology giving rise to
Show it to the camera.
to a conversion on humanity. And could we not have both, is what I'm basically asking. We start with a very fine-tuned cosmology, which then gives rise to a primordial Earth. That then, through evolution, gets a kind of biodiversity evolution, and then eventually you get a second stage of evolution, which is then this conversion to the talking about.
Well, yes and no. So I think that one idea is you have the beginning of the universe, you have the Earth coming. You get this biodiversity, but the diversity hasn't kind of converged later. It isn't like it first became very diverse and then it converged. It's continued. Diversity is continued on and on and on and on. It's just that the idea of Simon is that diversity was inevitably going to end up with certain types of complex organisms, including sentient ones like ourselves. But it's not as if it first became very diverse and then converged. Although there are ideas that maybe another way of looking at your argument is there is the idea that throughout evolution there are periods of stasis when not very much changes and periods when things change very, very rapidly, which is what we see. And the idea is that that's when a lot of novelty is generated, like in the Cambrian explosion, which then fights itself out in certain directions.
So would it be fair to say that through this biodiversity there isn't just one kind of spike that's pointed towards, but there are lots of little ones?
There's huge amounts of biodiversity, right?
Yeah.
There's been many, many different kinds of species over the history of life, millions of them. And there's lots of biodiversity now, which is one of the valuable things that we've tried to preserve. But it's not as if I should be careful. It's not as if we first had all this diversity coming out, which then kind of somehow became less diverse and channeled itself. That's not what we're saying.
I mean, also, Ard, is it worth pointing out that early evolution is actually non-Darwinian anyway, probably because of lateral gene transfer. And there's a quote, you had somebody there from 1999 that was definitely wrong. I mean, in the sense of what they were claiming, that early evolution was Darwinian. I think all we know about bacteria and the way they transfer information laterally, gene transfer, would suggest that evolution was much more Lamarckian in the early days. Do you want to comment on that a little bit? And,
yeah, I should. So that definition of life from Joyce was still used as saying Darwinian. I think Darwinian, it just means that somehow there was some kind of I'm a bit more flexible to use Darwinian. I just think I use Darwinian as being things that change over time and adapt themselves. And whether it has to be in this very particular neo-Darwinian way, I'm not so sure. Bose and Goldenfeld have written their papers partially on this topic. And the reason for that is this really striking piece of biology, which I'll point out to you. Which comes from Bose. So you can look at the genetic code. Okay. Genetic code is basically you've got 64 codons. So you've got these. Every 3 codons code for one amino acid. You've got 64 different ways of rearranging them, but you've only got 20 amino acids. So there's a redundancy between the code and the number of amino acids. And so you can look at things like if you have an error in that code, you have a mutation, it'll change one of the amino acids in the protein, and that can be really dangerous and not so dangerous. And Mose back in the 1960s said, well, there's something called the polar requirement, which says that if you've got, roughly speaking, you've got hydrophobic and polar amino acids, and if you've got a hydrophobic one, you want the error that you make to give you another hydrophobic one, which is not so different from the one that you had before. And if you look at, you can do the bioinformatics quite carefully, you can look at all possible ways of coding. And then it suggests that the human genome code is highly optimized for this polar requirement. And that's very surprising. Why it's 1 in a million? Why is it so highly optimized? And similarly, you can look at a number of other properties of the human genome, particularly a very interesting paper by actually Uyalon again, where you can point out that it's been also optimized for its ability to encode other things simultaneously. So the human genome doesn't just code, the proteins don't just code for, the genes don't code just for proteins, but they can also code sometimes for a number of other properties of the RNA. And it turns out the code is highly optimized for its ability to have other codes on it as well. And so the question is, how did this come about? And their argument is that this is through widespread lateral gene transfer. So the early evolution, you had these different genetic codes competing with each other. And eventually one of them won, and that's the one that we have. It's a very interesting idea. It's speculative. But I would still say that all the lateral gene transfer, I would still call it Darwinian in a sense. But even more, that's maybe perhaps a question of language. I would still say, maybe I could say this, it's still a stochastic optimizer. It's optimizing in this stochastic space in one way or the other.
It's just the origin of the diversity in this case is coming by a different mechanism. It still has to be selected for, and that's the still have the selection mechanism, which is key, and that would justify the use of Darwinian in that sense.
Could you just elaborate what you meant by lateral gene transfer?
Oh yeah, sorry.
Any other?
Okay, so that's just a fancy word. So basically, a lot of simple organisms like bacteria, they borrow DNA from each other.
Right.
So in fact, for example, a lot of antibiotic resistance. So when you get a lot of that is actually borrowed from soil bacteria, and so they go around. They do this all the time. In fact, they even use viruses, phage viruses, to keep like a repository of this DNA. They inject it back and forth. It's a very clever way of generating diversity. So what you might say is lateral gene transfer is just another mechanism, yet another collective mechanism, by which you can change yourself in very interesting ways. You can add and subtract modules. It's a very powerful way of your bacterium of changing.
Okay, well, lunch is just arriving, which seems to be a very good suggestion to wrap up. And first of all, to thank Art very much for a great talk. Thank you.